Dr. Look, unmute yourself.
Well, good afternoon. I'm Thomas Look, and it's my pleasure today to introduce our speaker, who is Dr. David Largispada, who is now the American Cancer Society Research Professor at the Cancer Center in the University of Minnesota in Minneapolis. And Dr. Largispada has a long career in cancer genetics. He started off his PhD training at the University of Wisconsin in Madison in the McArdle Laboratory, studying tumor virology in really a very fertile environment for these studies. Howard Temin was on his PhD committee and had a very productive training there, and then went on to join the group of Neil Copeland and Nancy Jenkins at Frederick within the National Cancer Institute. And of course, this effort was focused on insertional mutagenesis using retroviral vectors in the beginning to identify genes involved in cancer and vertebrates, of course, in mice, but also it turned out almost all the genes developed by this, or identified by this strategy, were also involved in human cancers, usually of the same histology that it was identified in the mouse. And so after a very productive training period during his postdoc, David went to the University of Minnesota, where he's been now for 25 years. And he started out, again, working on this very fertile area of insertional mutagenesis to learn about cancer genetics. And that evolved into studies of transposons as a means to identify genes whose loss or overexpression was involved in carcinogenesis in vivo. And he's progressed, of course, through the ranks. He's now a professor of pediatrics and genetics and cell biology. He's associate director of basic science in the Cancer Center. So it's really a pleasure to welcome David for this lecture. And his title is CRISPR and Transposon-Based Screens for Cancer, New Drug Targets and Synthetic Vulnerabilities in Sarcoma. David.
Thanks so much, Tom. Appreciate the chance to talk to you guys today about the work that we're doing at the University of Minnesota. This slide just shows my disclosures, and I've been involved in starting some biotech companies and advise a company called Recombinetics that I'll briefly discuss some work I'm doing with them. Some of our work is funded by Genentech. I'll briefly discuss that. You know, the motivation for getting involved in cancer genetics and identifying cancer genes is that, of course, identifying cancer drivers has had lots of implications for therapy and the idea of precision medicine. The problem is that some forms of cancer typically don't have many or any actionable alterations, and connective tissue tumors called sarcomas fall into that class. Some do have druggable target alterations like TRK kinase fusions or activation of the KIT tyrosine kinase mutant. More common are potentially druggable genetic alterations, but many genetic alterations in sarcomas are currently undruggable, and this would include the recurrent translocations that characterize some sarcomas like Ewing's sarcoma and alveolar rhabdomyosarcoma, as well as the genetic alterations that we see in p53 mutant genomically complex sarcomas like osteosarcoma and malignant peripheral nerve sheath tumors. So in my lab, we've been interested in how is it that we could treat cancer that doesn't have druggable genetic alterations based on today's knowledge. And so one approach that we've taken in my lab is based on the idea that there are useful drug targets that might be found by understanding copy number and epigenetically altered oncogenes and tumor suppressor genes. We're also interested in the idea that some of these tumors do have recurrent loss of tumor suppressor genes that could open up avenues for treatment based on the concept of synthetic lethality. And finally, we're investigating the idea that some of these genomically complex sarcomas produce neoantigens that have been overlooked for therapy. But today I'm just going to talk about these first two approaches that we're taking. And the approach that we've taken, Tom alluded to already, which is that in order to understand these genomically complex sarcomas with lots of epigenetic changes and gene copy number alterations, we could do forward genetic screens for similar cancers using transposon mutagenesis in mice. And that's the approach we've taken. And what I'll tell you, I'll explain how this transposon mutagenesis approach works in mice and show how we've used it to implicate known pathways in new cancers, identify new oncogenes, and implicate certain genetic alterations in specific cancer phenotypes. So when I started my lab at the University of Minnesota, I was looking for an approach to do induce cancers and cause their progression in mice that would be similar to human cancer, but reveal the genes altered during the process of tumor development. And our inspiration for this was work I'd done as a postdoc, where we were studying cancer induced by retroviral insertional mutagenesis. So certain retroviruses could induce leukemia or mammary tumors in mice by acting as insertional mutagens, typically causing the activation of endogenous proto-oncogenes by enhancer mechanism or by promoter insertion. The problem was there were no retroviruses available for inducing some of the types of cancer that I wanted to work on. And I was lucky that I came to the University of Minnesota, and a newly developed transposon system called Sleeping Beauty would function as a tool for this. This is a synthetic cut and paste DNA transposon derived from inactive TC1 Mariner family transposon elements in fish. And it was created in Perry Hackett's lab using a reverse evolutionary approach. So it's a two-part system. And you can think of it that way as composed of part one, the transposase enzyme, and the transposon vector DNA. And if you put both parts of the system in the same cell, the transposase recognizes inverted terminal repeats, excises the element, and causes it to reintegrate at a different position at a TA dinucleotide. So we could use this, we realized that we could use this system for somatic cell mutagenesis in mice, and it might provide the tool we were looking for. So we made one line of mice that carried multiple copies of a mutagenic transposon vector, and we integrated this as a multi-copy concatemer, depending on the line, anywhere from 25 to 300 copies, so that any one cell or clone of cells could acquire multiple independent transposon insertions. And we crossed those to mice expressing the transposase enzyme to get a mouse that has both parts of the Sleeping Beauty system. And in such a scenario, many, many somatic insertion mutations accumulated. We also designed the transposon vector so that it was insertionally oncogenic. So this was inspired based on what we knew about how retroviruses could cause cancer in laboratory mice. So we included a strong promoter and an artificial exon with a splice donor as a sort of an overexpression cassette. We included splice acceptors in both orientations and a bidirectional polyadenylation site as a disruption cassette, so that that by insertion, this vector could cause gain of function activation of proto-oncogenes or loss of function inactivation of an endogenous tumor suppressor genes. So depending on where and in what orientation it integrated with respect to an endogenous gene, you could get the expression of transcripts initiated within the transposon and splicing to downstream exons and cause overexpression of an entire open reading frame or just part of an open reading frame if an internal start site for ATG was used as a start site for translation. And the transposon could knock out a gene in either orientation. And again, we're mobilizing these transposons from a donor site that carries multiple copies of the transposon vector. one good thing about using Sleeping Beauty for this type of work is that it shows a relatively neutral insertion site preference to other mutagens that might be used. And this is work from Nancy Craig's lab, showing that with regard to various genomic markers like genes locations or DNase hypersensitivity sites, Sleeping Beauty integration doesn't show much of a difference from neutral compared to these other insertional mutagens. So to make a long story short, basically Sleeping Beauty worked for this purpose. And this just shows the pipeline for genetic analysis of Sleeping Beauty accelerated tumors. So we induce the tumors in mice, prepare DNA from those tumors, and then use ligation-mediated PCR to amplify the integration sites where the transposon is landed. And we do this with barcoded primers so that we can use next generation sequencing and pools of amplified DNA to obtain the unique insertion sites from each tumor, and then work with the bioinformaticists to distill this information down to the good stuff, the common transposon insertion sites. So this is like making whiskey from a lot of genetic data. So what we're looking for is the presence of transposon insertions from multiple independent tumors in or near the same gene. So if we sequence insertion sites from many tumors of the same kind from different mice, we're looking for these common insertion sites. And those reflect selection for rare insertions that have occurred by chance that affect the right proto-oncogene or tumor suppressor gene so as to give that cell a selective advantage and cause cancer. And when we identify these common insertion sites and the associated genes, it's possible to guess whether the gene is a tumor suppressor gene candidate or an oncogene candidate based on the position and orientation bias of the insertions. So this is some real data from a screen we did for gastrointestinal tract tumors. And insertions in this gene called WAC are spread throughout the gene with no strong orientation bias. And this reflects selection for knocking out WAC, which turns out to be a tumor suppressor gene. And here's a proto-oncogene candidate, arspondin-2, where the insertions were all in the promoter region of arspondin-2 and almost all of them and the same transcriptional orientation as arspondin-2, and they all turn out to make fusion transcripts that result in RSPO2 overexpression. So just the history of this is that we first published body-wide mutagenesis screens in 2005, and by 2009 we were publishing screens by doing tissue-specific mutagenesis with the SB system. And so the way that this is typically done is we use a CRISPR P-regulated form of the Sleeping Beauty transposase, where the transposase transgene's been knocked into the ROSA26 locus and can't be expressed until Cre recombinase removes a stop cassette, and then you get an expression of the transposase enzyme. And so by using different tissue-specific Cre drivers, you can mutagenize different tissues in the mouse and induce different types of cancer. And so this year, postdoc and I published a review on transposon mutagenesis for cancer genes. And there's now been over 65 papers on this, reporting genes for carcinomas, sarcomas, other forms of cancer. Many of the screens are done in cancer predisposed mice that express an oncogene or have a tumor suppressor gene mutation in the background. Screens have been done in cancer promoting environments like fatty liver, hepatocellular carcinoma. And now screens are starting to be published where the transposon mutagenesis is turned on to promote drug resistance or metastasis to get information on how that happens. In sarcoma, many different forms of sarcoma have been investigated using Sleeping Beauty mutagenesis, including two I'll talk about, osteosarcoma and malignant peripheral nerve sheath tumor that we've worked on. So I want to talk about this malignant peripheral nerve sheath tumors as an example of how the transposon mutagenesis implicated known pathways in this disease. These are Schwann cell lineage tumors. It's an aggressive, poor prognosis, soft tissue sarcoma that's chemo and radiation resistant, and we desperately need new therapies for MPNSTs. Half of all MPNSTs occur in patients who have neurofibromatosis type 1 syndrome or NF1 syndrome. They carry germline heterozygous loss of function mutations in NF1, an important tumor suppressor gene that encodes neurofibromin, a GTPase activating protein, and negative regulator of RAS. And so in these patients, when they develop tumors, it's because a rare cell has lost a wild type copy of NF1, and now you have a cell that doesn't express neurofibromin or any functional neurofibromin. You get increase in prolonged RAS activation in response to various stimuli and predilection for cancer. So when MPNSTs develop in NF1 patients, it's because of the stepwise process of histological progression and genetic changes. So loss of the NF1 wild type allele can lead to a plexiform neurofibroma, which is benign. Loss of CDKN2A and B causes the development of atypical plexiform neurofibroma that's thought to be premalignant, and then that can further evolve to malignancy by loss of the polycomb repressor complex 2. And this is due to biallelic loss of SUZ12 or EED, both of which are critical components of the polycomb repressor complex. So you can think of development of MPNST as like the sequential loss of barriers to transformation. First NF1, then CDKN2A or B, and B, SUZ12, and you get this progression. But it's not known if that's everything that's happening. And there might, we speculated there might be other changes occurring. And we performed a Schwann cell mutagenesis screen in mice using Sleeping Beauty, where we created mice where we could express the Sleeping Beauty transposase and mobilize the transposon that I talked about earlier in Schwann cells. We also did this on a p53 mutant background. We did it on a background overexpressing human EGFR as well, and in mice that both overexpressed human EGFR and had this p53 mutation. And when these mice were aged, they developed nerve tumors, some benign looking and some higher grade in a variety of nerves in the body. And so we took these benign tumors and the higher grade peripheral nerve sheath tumors and identified those common insertion sites that I explained earlier. And we predicted whether the genes identified were candidate proto-oncogenes, like FOXR2, which I'll discuss later, or were tumor suppressor genes. So one of the genes we saw with high frequency were insertions in NF1, and you can see the insertions are scattered throughout the gene, as you would expect for a tumor suppressor gene. We did pathway analysis on the genes we identified compared to genetic alterations in human MPNSTs, and also looked for cooperating mutations. And to follow up on this screen, what we've done is to knock out these candidate genes in human immortalized Schwann cells and malignant peripheral nerve sheath tumor cell lines. And we did this by making our own focused libraries, 2 to 4 guide RNAs per gene, and knocked out the candidate tumor suppressor genes in normal immortalized human Schwann cells, and knocked out the candidate oncogenes in MPNST cell lines, and looked for changes in anchorage-independent growth, transwell migration, and tumor formation in mice. And this just shows some of the data where we identified genes that when we knocked them out would confer upon these Schwann cells the ability to grow in an anchorage-independent manner. So they don't do that by themselves, but when we knocked out some of these candidate genes, we saw an increase in this behavior and ability to form tumors in mice. And the basic upshot of this is that we found out by doing this that sonic hedgehog, hippo-YAP, and Wnt beta-catenin pathways are enriched. The regulators of these pathways are enriched in the genes we identified and that we could validate in human Schwann cells. And they're altered by various routes in MPNST. And the reason it hadn't really been noticed that much is that depending on the MPNST, it can be a different gene. So for example, some MPNSTs lose GDI2, which we showed can activate hippo-YAP signaling in Schwann cells, or they lose NF2 or tau kinase 1 that also regulate hippo-YAP pathway. Any of 3 different members of the destruction complex for beta-catenin are downregulated in MPNST, or you have overexpression of RSPONN2 in a subset of MPNSTs. So now we can add other pathways to this idea, this stepwise loss of tumor suppressor genes that are involved in ultimately making an MPNST. And this information gives us new treatment strategies that we could pursue for NF1 or for MPNST. So if all of these tumor suppressor genes are lost in different subsets of MPNSTs, they suggest other pathways that are altered and targets to attack with therapy. So obviously when NF1 is lost, RAS-activated pathways like MEK might be inhibited. When CDKN2A is lost, CDK4 or 6 inhibited. As I'll show you, if polycomb repressor complex 2 is gone, the cells become sensitive to HDAC inhibitors. With patched smoothing inhibitors, loss of PTEN, PI3 kinase inhibitors, and for the Hippo-YAP pathway, perhaps TEAD inhibitors, which are being developed now. So that's really one story about how transposon mutagenesis has helped us. Another example is that it's helped us identify new oncogenes. And one that I think is particularly interesting that we identified in this screen is FOXR2, candidate proto-oncogene in Schwann cells. So a subset of the tumors that we identified had transposon insertions in the promoter region of FOXR2 or an intron that created fusion transcripts, all of which, when translated, would result in overexpression of the entire open reading frame of FOXR2, which is contained in a single exon. This is a gene that's located on the X chromosome. And when we started working on it, not much was known about it. It has a fork-head box domain, 2 low complexity regions, putative nuclear export signal and nuclear localization signal. Later, others found that there's a region that binds to c-MYC. More on that in a minute. We found that a subset of MPNSTs express FOXR2 at high levels, and that might be because this region of the X chromosome is amplified in many MPNSTs. And so this is an example of a gene we wouldn't have picked out easily just by studying human tumors. And so a variety of data support FOXR2 as an oncogene. We and others have identified it by transposon mutagenesis in a variety of types of cancer and published some of that. In 2016, FOXR2 activation by chromosomal translocation was reported in a subset of central nervous system primitive neuroectodermal tumors in the brain by a group in Germany. And a group at MD Anderson reported it as a breast cancer oncogene identified by computational approaches. And so it's a member of a large family of transcription factors that has lots of different biological roles, including in embryonic development. These can act as pioneering transcription factors that make it DNA accessible to other factors, and there's 35 plus members in mammals. And the defining feature is this fork-head box that is a winged helix domain that binds to DNA. So we overexpressed FOXR2 in human Schwann cells and did RNA-seq analysis and saw that there was upregulation of MYC target genes. Interestingly, one of the MYC transcript level itself was not altered by FOXR2, but MYC protein levels are altered when you overexpress FOXR2 in human Schwann cells, as shown in this Western blot. And if you knock out FOXR2 in an MPNST cell line that expresses it, you reduce MYC protein levels. So we went on to show that FOXR2 can bind to c-MYC by co-immunoprecipitation assays. It also binds to N-MYC, but not L-MYC. And that's basically what these data show. FOXR2 expression at high levels can stabilize MYC protein. And so in these experiments, we measured MYC levels at various time points after adding cyclohexamide. And you can see in human Schwann cells expressing a lot of FOXR2, the MYC half-life is very long, whereas it normally has a very short the MYC protein normally has a very short half-life in these cells. Now, the group at MD Anderson had shown some of these same results and identified the region of FOXR2 that binds to MYC and created mutants that of FOXR2 that wouldn't bind MYC. And so we mimicked what they did and showed if you delete that domain, sure enough, you no longer IP MYC with this FOXR2 MYC mutant, but it still can transform cells in vitro and in vivo. So this is soft agar colony formation. This is tumor formation in vivo with a mouse neuroblast cell line, but the same results with human Schwann cells. And this is just more data showing that if you get rid of the MYC binding domain, you still have a protein that can cause transformation, just not as efficiently as the full-length protein. And we found out that there's another function for FOXR2, and it promotes focal adhesion kinase and SRC activation, and it does so in a MYC-independent manner. So in mouse neuroblast cells where we overexpress FOXR2, you see activation of focal adhesion kinase. If you knock out FOXR2 in a human medulloblastoma cell line, it reduces the activation of FAC, which you can restore by putting FOXR2 back in. And the interesting thing is that if you express this form of FOXR2 in cells that can't bind to MYC, it still activates FAC. So we think it might have two functions. So we asked the question of whether FOXR2 could be involved in human cancers and besides the CNS PNETs that have been described earlier. And if you just look at copy number versus expression level for FOXR2 in human cancer cell lines, you can see that it is expressed in a subset of cell lines. In some cases, that is associated with the increased copy number for FOXR2, like in MPNSTs. So a subset of neuroblastomas express, and they're highlighted here in yellow, express FOXR2 at high levels, or at least neuroblastoma cell lines, and a subset of gliomas, sometimes with copy number alterations. So we got interested in FOXR2 in neuroblastoma and working with John Maris at Children's Hospital of Philadelphia. So showed that FOXR2 is expressed at high levels in a subset of human neuroblastomas that lack NMYC amplification. And this subset also has high risk and poor prognosis. If you look at survival in this subset that has high FOXR2 levels, it's really poor, like the NMYC amplified group. So it defines a new subset of poor prognosis neuroblastomas. And in the Maris lab, they showed that this subset of FOXR2 high non-MYC amplified neuroblastomas are associated with the core MYC core gene expression signature that you also see in MYC amplified cases. FOXR2 is expressed at high levels in non-MYC amplified neuroblastoma cell lines, and we showed is activated by fusion with another gene called KLHL13 in a cell line called SKNAS, a well-known human neuroblastoma cell line. So this fusion is the result of a chromosomal inversion on the X chromosome in this cell line that juxtaposes the first exon of KLHL13 with exons of FOXR2, and this fusion transcript overexpresses FOXR2 in the cell line. And if you look at non-MYC amplified human neuroblastoma cell lines compared to MYC amplified and MYC amplified cell lines, generally there's dependency on FOXR2 in these non-MYC amplified cell lines. And if you knock down FOXR2, that destabilizes c-MYC in these cell lines as we would have predicted. So this is an interesting oncogene that might function as encode an oncoprotein in two different compartments in the nucleus, where it binds c-MYC and n-MYC and increases their stability, and it increases MYC-dependent transcription, and perhaps also in the cytoplasm by an unknown mechanism can activate focal adhesion kinase and SARC. We think this is a really good drug target in cancers that overexpress it, because it's normally only expressed in the testes postnatally. And so in tumors that are FOXR2 high, and you have transformation due to effects on MYC, you could target this interaction or try to destabilize FOXR2, and it would be a way of targeting MYC function. So with regard to transposon mutagenesis, I want to tell you about how we can use it to establish genotype-phenotype correlations. So in cancer, we have really complex phenotypes and genotypes in human cancer, and a lot of fancy machine learning and other informatics is used to try to tease apart the causal features for patient phenotypes. And with transposon mutagenesis, we're kind of interested in the idea that we can make things simpler, like fruit fly genetics. And so I'll show you how we can use transposon mutagenesis to implicate certain genetic alterations in specific cancer phenotypes. And this is all related to the fact that transposon mutagenesis is designed to affect endogenous genes in ways that create fusion transcripts. So I said earlier, when transposon mutagenesis activates an endogenous proto-oncogene, it's often because it creates a fusion transcript initiated within the transposon that results in ectopic expression of downstream exons, like with FOXR2, or it may cause overexpression of a truncated transcript, or it can disrupt a transcript in either orientation. But all of these events create fusion transcripts that you can detect by RNA sequencing. The good part about doing RNA sequencing on Sleeping Beauty accelerated tumors is that you could also study their gene expression profile. And so when we do this RNA-seq-based analysis, we look to see what genes are altered by transposon mutagenesis, but we also look for clusters of covarying genes. So if we get a gene expression pattern like this, where you have genes along here and cases along the top, you can look for genes that covary together. And so for each cluster, for each tumor, you can get a single score for that cluster of genes that covary. And it turns out that this allows the definition of multiple independent transcriptional patterns within a single data set. And these clusters of covariant genes often are very meaningful. So some of them, for example, are genes that go up and down with the cell cycle. And so you can define the relative cell cycle activities in a set of tumors. Some of them reflect infiltration by immune cells. And so you can tell by RNA sequencing if the tumor was immune hot and had a lot of immune cells in it or immune cold. And so if we do this RNA-seq on Sleeping Beauty accelerated tumors, we can get the gene expression profiles, and we can infer the phenotypes from that gene expression profile. But we also find the driver genes at recurrently altered that are as recurrently altered genes, transposon fusions. And so we can put this information together and try to find genotype-phenotype correlations. So I'll show you an example that we validated with osteosarcoma. So we did a screen for osteosarcoma on a p53 mutant background. A subset of these animals developed metastatic disease. And we found NF1, NF2, PTEN, and other tumor suppressor genes. We found oncogenes like colony stimulating factor 1 receptor, SEMA4D, and others. And this shows some of the genes that we found. So it included a lot of tumor suppressor gene candidates and proto-oncogene candidates. And we noticed that in these mouse tumors, some were immune hot and some were immune cold. And you see the same thing if you look at human osteosarcoma and canine osteosarcoma. And we compared transcriptional analyses in these osteosarcoma from these three species and reported it in this paper in Cancer Research. This shows an example of the immune cell clusters and some of the genes involved in human osteosarcoma, in the mouse osteosarcomas, and in the dog osteosarcomas. And you can see how they co-varied. And that gives you some information about the immune cell infiltration status of that tumor. And this is important because whether the osteosarcoma is immune cold or hot is prognostically significant in human osteosarcoma. So it turns out that if you look at survival versus time or metastasis versus time, if the primary tumor has white blood cells in it, and that's these cases marked with the red line, that's a good prognostic indicator. Okay, even though the primary tumor is removed by the surgeon, it may reflect the fact that somehow the immune system has recognized or reacted to the tumor. So this is an important phenotype that we'd like to understand better, and pathways that control it. So if you look at the immune summary score in the mouse osteosarcomas accelerated by Sleeping Beauty mutagenesis, it turns out that among the ones that have the lowest immune summary score, there are some tumors with T2-onc CSF1 receptor fusions. So if the osteosarcoma has activation of CSF1 receptor, now you have an immune cold tumor. Now, this encodes the cytokine receptor for IL-34 and macrophage colony stimulating factor. When CSF1 receptor is expressed at high levels on cells, it can be shed and may be acting as a decoy. We don't really know. But we did create mice where we overexpress CSF1 receptor on a P53 mutant background, and it definitely accelerated osteosarcomagenesis compared to mice with just CSF1 receptor overexpression or just P53 mutation. And it turns out that these faster developing osteosarcomas expressing CSF1R compared to controls osteosarcomas do have a relatively immune cold phenotype where there are fewer immune cells infiltrated into these tumors. And so here's an example where we found a driver of osteosarcomagenesis in this mouse model that's associated with an important phenotype. Now, we don't know how CSF1 receptor expression on the osteosarcoma cells causes this phenotype, and it may not do the same thing in human osteosarcoma, but it gives us an entree, a molecular mechanism we could pursue now to study this interesting tumor phenotype. And we've done the same thing in some of our other Sleeping Beauty screens. So this is UMAP projections of mammary tumors induced by Sleeping Beauty mutagenesis in mice. And these UMAP projections, show transcriptional profiles. And basically, we found out that there are 5 different molecular subtypes of mammary tumors induced in the Sleeping Beauty model, and they correspond to different PAM50 subtypes of human breast cancer. And the genes mutated within these clusters differ. So these clusters here tend to have PTEN inactivating insertion, whereas this cluster here has insertions that activate plaqueglobin, which is encoded by the JUP gene, and they express an N-terminally truncated form of JUP. And it turns out that that same cluster is low in estrogen receptor dependent gene expression and has a very low immune score and high cell cycle. So here again is an example where we can associate a specific genetic alteration with some interesting tumor phenotypes. And that's something we're pursuing now. So the next story I want to tell you about has to do with this idea of looking for synthetic drug sensitivities, because the tumors that I'm talking about may not have many obvious activated oncogenes, but they do have certain tumor suppressor genes that are recurrently lost. And I told you about the MPNSTs, how they seem to develop from the sequential loss of certain tumor suppressor genes. And so we've created human immortalized Schwann cells that lack NF1 or lack NF1 and SUS12, which is required for polycomb repressor complex 2 function. This Western blot just shows how we were able to knock out SUS12, and that completely eliminates histone H3K27 trimethylation. That's the chromatin mark that the polychrome repressor complex 2 makes on histone H3, that lysine 27. And using these cells and screening drug repurposing libraries, we've been able to find drugs that show selective toxicity to NF1 deficient cells or SUS12 deficient cells. And this shows you some examples of the drugs that we identified. So for example, when NF1 is gone from human Schwann cells, they become strikingly sensitive to cardiac glycosides such as digoxin, tubulin depolymerizers, and MEK inhibitors, which maybe isn't so surprising. When polychrome repressor complex is gone, the cells become hypersensitive to histone deacetylase inhibitors, DNA methyltransferase inhibitors, and aurora kinase inhibitors. So this gives us potential drugs we could use. We started to combine these drugs to look for synergies. And this just shows an example of one synergy that we've been pursuing, which is if you inhibit histone deacetylase and MEK inhibitor, it shows synergy in vivo versus MPNST xenografts. And this is an example of a cell line xenograft. You can see percent survival versus days after enrollment of these tumors, of these mice when tumors reach 250 millimeters cubed. And so without any treatment, the mice have to be euthanized quickly because the tumors grow out of control. But if you combine these two drugs, vernistat and selumetinib, an HDAC inhibitor and a MEK inhibitor, you can dramatically prolong the life of these mice. It induces apoptosis in these tumors, and it can shrink the tumors, unlike either drug alone. So this just shows maximal response in tumor volume. And we get the same results when we use patient-derived xenografts that we or others, our collaborators have created recently for MPNST. This just shows one particular example. Here's non-treated, and here's treated with the combination. And this shows that percent change from baseline. Again, the combination can cause dramatic shrinkage of the tumors. And same thing with the second PDX. So I've shown you how we can use CRISPR knockout cells to do drug screens to identify promising compounds for treatment that leverage loss of specific tumor suppressor genes like NF1 and SUS12, and how we can find new drug targets using Sleeping Beauty transposon mutagenesis as well. And I think we're in a position now where we have kind of too many drugs to pursue, and we're trying to figure out how to go forward from here. And so we've tried to invest in new model systems that would be predictive for MPNST treatment. So for example, we've been making human-induced pluripotent stem cell-derived models of NF1-associated peripheral nerve sheath tumors. Working with a company, we created and characterized a porcine model of NF1 syndrome that develops at least benign peripheral nerve sheath tumors. We've created MPNST patient-derived xenografts, and we're part of a consortium working on this with Angela Irby at WashU in St. Louis and Christine Pertillis at Johns Hopkins University, and created next-generation genetically engineered mouse models of MPNST. But the last story I wanted to tell you about is how we've used these MPNST patient-derived xenografts, both in vivo, but also created a new in vitro drug screening approach that uses something called 3D microtissues, developed here at the University of Minnesota by David Wood. He is in the Department of Biomedical Engineering here at the University of Minnesota. And he's developed an approach where you can embed cells, including, as we started to work on together, primary PDX cells in a collagen solution and create little microtissues. And you can do this in such a way that you can determine how many cells you want in each microtissue and transfer these to well for an experiment. And the good thing about this is that you can determine whatever cell types you want to put in here. You can put in tumor cells or tumor cells and other cell types. You can vary the components of the extracellular matrix. And here's what some of these microtissues look like. So you can create them in a very uniform manner. Here's some real pictures of microtissues that we made with cells from different patient-derived xenografts. And when we do this, we remove all of the dead cells and we remove the mouse cells. So we're just putting the human tumor cells in here. And we can culture them for a week or more for the good PDXs anyway, and then do live dead stain and count them and expose them to different drugs. So working with this team, we've created and characterized now over 20 MPNST PDXs because there weren't many in the field. And about half of them we've tested in the microtissue platform. Some of them do not form robust 3D microtissues and some do. So that turned out to be a characteristic feature of each PDX that doesn't change. Some grow robustly, some are good, and some are unusable. And it turns out that the gene expression profile of the PDX that produces good or robust microtissues is actually different from the ones that produce poor quality, unusable PDXs. We don't know what this means, but it's a biological feature of these tumors. And so the way we do this is to propagate the PDXs, remove the tumor, remove mouse and dead cells, generate the microtissues. They're plated in a printed microwell array, and we can test the effect of a drug on several dozen microtissues at once. That's typically what we do, and count the live and the dead cells after drug exposure. And so we can take these PDXs and we can find out how they respond to chemotherapies. So here's just an example of one PDX we made in a bunch of different response to a bunch of different chemotherapies at different drug concentrations. We can do MEK inhibitors, mTOR and PI3 kinase pathway inhibitors, different RTK inhibitors, and other drugs. And by doing this, what we've started to find out is that PDXs, they respond differently to the same drugs. So here's 3 different drugs, 2 MEK inhibitors and a PARP inhibitor, and with 4 different PDXs, and they vary in the way they respond to these drugs, and the maximum percent killing is different. IC50s are different. So this represents a challenge, I think, for drug development. We're also starting to combine drugs in these 3D microtissues. And for certain combinations, like selumetinib and digoxin, we see evidence of synergy where the combination can kill at doses that the 2 drugs, when combined, at doses that the 2 drugs singly don't do anything to. And same with selumetinib and vernistat. So those are that's a combination I showed you already. This combination actually works very well too. And so now we've started to generate tons of data on a variety of drugs from different PDXs, and we're starting to see patterns. So this is all early data really, but I just wanted to show it to you as sort of an evidence of a way forward when you have a lot of drugs that might work. And you're wondering what combinations would be best to pursue in vivo studies with the PDXs, this might be sort of an intermediate step. So just to conclude, I've shown you how transposon mutagenesis reveals activated pathways in MPNST due to tumor suppressor gene loss. It identifies new oncogenes that activated in a subset of many human cancers. And I gave you FOXR2 as an example, but I think there are others that we've identified as well. It has also helped reveal drivers of complex tumor phenotypes like CSF1R and JUP and immune cell exclusion. And synthetic drug sensitivities might also provide new drugs useful for MPNST. And we're hoping that PDX cells growing in these 3D microtissues is sort of an intermediate stage tool for us for evaluating drugs and drug combinations. So these are the people in my lab that were involved in all of this work, and we've got many great collaborators here at the University of Minnesota and elsewhere, and this is the source of funding for this work. And I'm going to stop now, and I'm happy to take any questions that you might have.
Great. Can you hear me, David?
Yes.
I think I'm back on. So thanks for a great talk. It's really, you're really making terrific progress with all these models in defining the molecular pathogenesis of particularly MPNSTs. I wanted to ask you, I didn't know that the YAP-TAZ pathway was implicated. And could you remind me again how it's activated?
Yeah, so any of a number of negative regulators of hippo-YAP signaling are either downregulated or actually deleted in MPNST. But because it's not always the same negative regulator, it hasn't really been that apparent. So, for example, a subset downregulate NF2. A subset downregulate or delete tau kinase 1. And it turns out tau kinase 1, and these are known regulators of hippo-YAP signaling that people have found in other systems. Tau kinase 1 actually maps near NF1 and SUS12. And so it's sometimes co-deleted with those other tumor suppressor genes. So that's how it is getting, that pathway is getting activated in a subset of MPNSTs. So it could be GDI2 loss, NF2 loss, tau kinase 1 loss.
Great. No, I was just wondering, do you know if this occurs? Of course, the MPNSTs arise from a limited number of plexiform neurofibromas that each patient has after development. And do you know if the YAP-TAZ is already activated and stimulated the proliferation of these benign tumors, or is it part of the true malignant transformation?
It's a really great question. And we recently created a tissue microarray with plexiform neurofibromas and MPNSTs on it. And one of the things that we stained for was YAP, so we could look for nuclear YAP. And the stain's been done, but I don't have the results yet. So I don't know. But I do suspect that the high interstitial pressure caused by the HA-rich the hyaluronic acid microenvironment within the plexiform neurofibromas might be activating the YAP pathway. And that is, we know that plexiform neurofibromas are hyaluronic acid rich. And in other tumor types, that HA richness and the high interstitial pressure can activate the hippo-YAP pathway, which is stiffness, is the pathway that's sensitive to tissue stiffness. So it's true in breast cancer. So I have this, kind of pet model system or model idea that in the benign stage, you have activation of the pathway by environmental factors. But then when you evolve to malignancy, it's by a genetic means. And so the cells become, activation of that pathway becomes independent of the microenvironment. That's the idea we're pursuing.
That's really fascinating because MPNSTs are very bad and relatively rare, but most NF1 patients have one or more plexiform neurofibromas, and those can be enormously troubling, even if they're not malignant, but they can be in bad locations and enormously troublesome for the patient. So that would be really cool if like TEAD inhibitors would work even earlier and help those patients as well. Well, we have a question here in the Q&A. We have two. So I'll read the first one from Nicole Solimni. Do PDX cells continue to proliferate in 3D microcultures? And have you tried any immunohistochemistry staining on these cultures?
Yeah, so that the answer to both questions is yes, but the PDX cells continue to proliferate in the microtissues only if it's one of the robust class, and that's like a subset of them, and about a quarter of them will do that, and that's good because some of the drugs that we're interested in have for their effects to become manifest. You know, we're told the drug has to be there for 5, 6, or 7 days. So for those, we can do that. For the ones that are just good, they survive but don't proliferate much. And then the unusable ones, the cells just die right away in the microtissue. Even though we tried different media formulations, some of them we can't keep alive. And one of the things that David wants to do is try other cell types and ECM components to see if we can get better survival. We also have tried immunohistochemistry, and that works just fine. You can do all kinds of IHC with these, and you can count the number of cells that went through S phase, for example, by detecting BRDU positive cells, etc.
Well, we have another question from Antija Voy Hartley. Several studies have shown that YAP-TAD inhibition in NF2 models only result in cytostasis, not cytotoxicity. That, of course, might not be ideal for use as a drug clinically. Any comments on this?
Yeah, I agree. We noticed the same publications. We're now working with the company that has a series of TEAD inhibitors, and it also looks like we see cytostasis. So our thought is that perhaps as a component of a drug cocktail, though, it could still be useful, and that's sort of what we're hoping with the 3D microtissues we can accomplish, is find out what the magic combination of pathway inhibitors would be, some of which might be just cytostatic, but when combined, you see cell death. So, and I actually showed an example of that where the HDAC inhibitor or the MEK inhibitor by themselves versus the PDX cause the tumor to sort of just stop growing, but when you combine them, you actually see tumor shrink and you see a lot more apoptosis.
So it might be possible to combine TEAD inhibitors
That's what I'm
with
saying. Yeah.
something else.
With something else, yeah.
Okay, well, are there any other questions for the Q&A box? Well, I think we're right on time here, so perfect time to end the lecture. Thank you very much, David, for a terrific talk. Really raised a lot of questions in this very enigmatic tumor, and you've made remarkable progress in working out the molecular pathogenesis. Thanks a lot.
Thank you, Tom. Thanks, everybody. All right.